I'm here again. Thank you. I was told to speak on spinal cord injury and neuroprotection, especially in relation to some nano delivery of drugs. And I'm happy to do that. This is not a very clinical experiment because we limited our investigation up to 5 hours at the moment because we wanted to study biochemical changes in the early hours after a focal spinal cord injury. And as usual, there are several observations, and these summary and conclusions are mine. We believe in the microenvironment of the spinal cord, and therefore we feel that whenever this capillary is leaky, whether in the spinal cord or in brain, it can affect both the glial cells and the neurons. And we also believe that the blood-CNS barrier, that also includes the barrier in the spinal cord, is crucial in producing edema, necrosis, ischemia, and all other gene expression leading to cell death. So this is our hypothesis, and we try to block at different levels using drugs and see that whether we can reduce cell damage. We are also doing some experiments in Army, yes, with the military hospital, and this is our collaborators there. And I must admit that we have very little. After doing lots of experiments, we still know very little. As I told you yesterday, that spinal cord injury is the major problem of war injuries. And these are quotes from various organizations telling that explosions are one of the main reasons for traumatic spinal cord injury. Rather, a large number of cases occurs resulting in explosion, and as you can see that our soldiers are very young, and therefore the time of injury largely occurring in young people. As you can see that 53% of all gunshot wounds to the spine resulted in a spinal cord injury, and you are more expert in the audience than me. These are some historical cases, a very interesting one, and I'm sure most of you know very well. This is Lord Nelson, and then this is US President, who even survived a long time after injury but could not be saved. I started working on a spinal cord injury and with multi-focus approach, also clinical neurophysiology, and Dr. Dimitrijevic here, and he is pioneering in spinal cord evoked potentials. I have pleasure to work with Dr. Erik Stolberg in Uppsala, and we came out with this book in early 1998 by Springer, and some of you, including Weisian, has given a chapter. We were very obliged to him. And I must confess that these observations, we are only trying to mimic some of the human cases, but obviously this is the problem because animal experiments cannot be replicated in any way in human situations. There are obvious regions, and one of the most important causes of a spinal cord injury is motor vehicle accident, apart from gunshot or explosion. But the point is that what we do in animal experiments, we always injure a spinal cord on the dorsal side. But in clinical cases, spinal cord injury occurs always on the ventral side. So these are the basic difference between experimental and clinical works. And obviously, in all experimental animals, we have to anesthetize them first. After that, we make injury. This is almost impossible in human cases because they are occurring in conscious situation. So these are the drawback in experimental situations, but nevertheless, we try to study some molecules that are involved in pathological process of spinal cord injury. So when we try to study the spinal cord, we found that there are several models, that transaction, crush and contusion, are available, and we wanted to study neuroprotection at cellular level. So we try to develop another model of spinal cord injury, and here we have a model here where we can make a lesion of dorsal horn 10 to 11 segment on the right side. The reason is that we wanted to study drug effect in short survival period, both the ipsilateral and contralateral side. So this injury model can be considered probably one of the new models in spinal cord injury at that time. And you can see here that we tested experiments, and here this spinal cord is much more better than the untreated spinal cord. There are also different drugs that we have applied, no matter what they are, but you can see we can differentiate different spinal cord with different drugs, so that we can understand that in a short survival of 5 hours, we are able to protect or at least reduce the changes or thwart the changes for some time, when the soldiers can be taken to the expert hospitals. So our idea is we cannot say that we are preventing the spinal cord changes, but we are stopping for a while so that the some changes are reduced while they are transporting to the hospital or something like that. This is the idea, of course, in experimental situations. Regarding spinal cord evoked potential, I can only show you very briefly that these evoked potential on the rostral side, we have recorded rostral and caudal both sides. The rostral spinal cord evoked potential amplitude disappeared, and there is a gradual increase in the positive potential and also shifting in the latency. Whereas the caudal side, even the amplitude has increased. And this is not surprising because you are experts here. When we cut some spinal cord or injury, there is always adding of inhibitory or excitatory potential. So it is quite simple that behind the lesion, below the lesion, we do not see damage on the evoked potential, but they are increased. Then, to study this function, we tried some drugs which are known to have neuroprotection, and some drugs which are not, because we try to understand. And I only show here the rostral evoked potentials, and we have different drugs, cyproheptadine, serotonin receptor blocker, works on this case. Dexamethasone in the doses that we have used do not show any good result, wherever indomethacin and ibuprofen showed very good results. On the other hand, propranolol and phentolamine did not show good results here. Nimodipine was one of the best. So what does it mean, the rostral spinal cord evoked potential amplitude and latencies? Whether we can predict some changes in the short period of time? And the answer is probably yes. When we compile these pathological changes, you can see that those drugs that produce neuroprotection are able to maintain rostral spinal cord evoked potential much better. We measured edema as a parameter up to 5 hours after injury and correlated with 2 minutes changes in the amplitude of the spinal cord evoked potential on the rostral side. We find a very tight correlation, indicating that at least up to the period of 5 hours we followed, the early evoked potential changes can be predictive of edema or swelling. So this is the pathological picture of a spinal cord up to 5 hours. You can see even there are changes in different sides, but this is much more swollen. Leakage of Evans blue is gradually higher up to 12 hours, and also we can see that there are differences in the rostral and caudal side in the magnitude of the leakage. This is the injured spinal cord area, and the far above the areas do not show leakage of Evans blue in this case. But when we have other tracer, the small molecule tracer, radioiodine, you can see we can detect changes in the spinal cord permeability as early as 5 minutes, and it is going up to 12 hours we have observed, but still the changes in the caudal and rostral side were different. As a pathologist, we always try to understand the passage of the leakage, and here I can show you that the leakage was not across the tight junctions, they are not widened, but it is the infiltration across the membrane. Here is one example, but not all cells are infiltrated. One cell is infiltrated with lanthanum, but the adjacent cells were quite normal. So this is a very specific phenomena, probably due to some chemical, neurochemical effects. We do not know. This is another example. After 5 hours, there is massive leakage of lanthanum. But when we give drugs, you can see we are able to produce this less infiltration, although the lanthanum can be seen in the vesicles. But the tight junction here is intact. So the point is that this is the membrane permeability of endothelial cells is increased after trauma, and these sections were taken far away from the physical lesion. So this is T9 level. It means some diffusion of chemicals are occurring, causing membrane permeability. Obviously, edema is also progressing, and we can still see the changes of edema in rostral and caudal differently. Neuronal changes we can see after 5 hours, quite severe, as well as we have severe glial changes, and it can also be differentiated between the left side and right side. Right side is the lesion, and this is the T9. Lesion was done in T10 and 11. So there is also differences in the sides. Myelin basic protein, here you can see that control versus T12 right side showing much more degeneration, and swelling is evident. At that time, people believe that pathology is a very long process. So what we are measuring at 5 hours, either it is artifact or something wrong. I mean in early 90s. To prove that, we have also studied immediate early gene expression, and you can see this is CFOS, both dorsal and ventral horn, they were upregulated. So these changes cannot be due to artifact as well. At electron microscopy, we also tested our hypothesis, and you can still see that T9 right and T9 left. Still we can see the differential changes. The right side show much more myelin vesiculation and edema as compared to the left side. So it appears that the model is working in our hand. But coming to the drugs, of course, there are lots of substances. They are released after spinal cord injury. Some of them are good, some of them are bad. So we tried neurotrophic factors because they are known to produce good results. And you can see here that this is untreated and T12. BDNF treated. When we combine BDNF plus GDNF, we can get much better results histologically. And then we treated with IGF-1. We apply topically over the spinal cord, and we can see that if these IGF-1 or BDNF were applied minus 30 minutes to 10 minutes, even up to 30 minutes, the results are better. But applying 60 minutes after, they are not good. It appears that IGF-1 in similar concentration may have better results. And this is also showing the leakage of iodine, the similar results. What could be the mechanisms of these neurotrophic factors when applying over the spinal cord? Do they change some molecules or working in another way, because 5 hours is too short for regeneration? What we can see here, that there is upregulation of neuronal nitric oxide synthase after injury that was significantly reduced by BDNF application and also IGF-1 application over the spinal cord during 5 hours. Some elongation of these actions can be seen. It means that 5 hours is also quite a good period. At the level of electron microscopy, BDNF application prevented leakage of lanthanum as compared to the untreated group, and also BDNF reduce the myelin degeneration here in this situation. We have studied nitric oxide, and also the other free radical is carbon monoxide. It is synthesized in the body by heme oxygenase 1 and 2. There is a lot of evidences that HO1 could be beneficial and HO2 is constitutive. So we just tried to see that what is happening on heme oxygenase constitutive isophore. Here you can see that this is massive upregulation of HO2, but PCPA, this is serotonin synthesis inhibitor, I showed you earlier was neuroprotective. We can see that HO2 expression is much less evident. So it means that those situations, when they produce neuroprotection, they have less expression of HO2 and neuronal nitric oxide synthase. Then together with Professor Huang, I think is not here unfortunately, we did some experiments in Beijing, and we have combined even more than 2 neurotrophic factors. So this is a combination of ciliary neurotrophic factor, BDNF, and glial-derived neurotrophic factor, a combination, and we applied over the spinal cord in the same model. And what you can see here, a combination of 3 neurotrophic factors was the best in reducing blood-spinal cord barrier permeability after spinal cord injury as compared to the other combination. This is same occurring with reduction in edema formation. And also the spinal cord local blood flow was higher with the combination of 3 neurotrophic factors as compared to the other combinations. Neuronal damages was also maximally reduced in our model. HO2 expression likely was also significantly reduced after the combination of 3 neurotrophic factors. Here is an example of these neurotrophic factors application on HO2 expression after BDNF, IGF-1, and I'm showing you this triple application of neurotrophic factors. We hardly see HO2 expression as compared to here untreated group. So we can say in this model that early application of drugs or neurotrophic factors within say 1 hour is able to reduce pathological changes up to 5 to 8 hours. And this is also the myelin, we have used nimodipine, and you can see that nimodipine has the best effect on reducing spinal cord pathology and myelin vesiculation as compared to the untreated group. This is another example at the electron microscopy. Nimodipine is best in experimental model in our hands so far. What is the relation between human cases of a spinal cord injury and animal model at short period of time? Byron Kakulus, as you all know, is having lots of spinal cord injury model in human and many samples we are using. So here we have used 6 hours of human spinal cord injured in I think T12 region. And we have studied leakage of albumin, and you can see that albumin leakage is quite extensive in human specimen of the spinal cord. This proves that probably leakage of blood-brain barrier to protein molecules are playing important roles. I do not say that 6 hours of human life and 6 hours or 5 hours in rat spinal cord injury could be similar or comparable, because rat's life is 2 years. But the point is that we can see similar changes with more extensive in human cases. So again, we believe that blood-spinal cord barrier or blood-brain barrier is very important. In last 5 minutes, I will show you, I promised to show some
second lecture also done?
no, later.
So please come to the conclusions.
Okay. So the conclusion is that application of drugs within a short period of time after injury can reduce or thwart the changes for some time. And we need to study more the combination and also the physiological functions. Thank you very much. I stop here.
Thank you so much. I'm really sorry. All these talks are so interesting that I enjoy it completely. But anyhow, we have a little bit to stay in our time. And now I would like to ask our next speaker, Dr. Hang Yun from China, detailed interpretation of consensus for clinical neuroprotective progressions in patients with complete chronic spinal cord injury. And I would like where is the speaker? So